Okay, so now we get to Mendel's second idea. Mendel wondered, does one trait affect another? Because just observing people and pine trees and things, it seemed like some traits go together. Red hair and freckles is a common one. It seems like they go together when they get passed down from parent to offspring. Does one affect another, or are they independent of one another? Well, to do that, to figure that out, you now had to keep track of at least two traits at once. So the crosses we were talking about with his pea plants before, when he made it his pea plants, he was only looking at one trait. We call those monohybrid crosses. It simply refers to how many traits are you choosing to look at. Well, now he chose to look at two traits at once, right? So he had, I'll use the example on the next page, I think. He did the same series of crosses. He started with pea plants that were pure dominant, this time for two traits. The example that they use here in this PowerPoint slide has seed color and seed shape. Yellow seed color, yellow peas is dominant. That's just the way it is. Round peas is dominant. Green peas is recessive, and wrinkled seeds, wrinkled peas is recessive. So first he developed a group of true breeding dominant ones for both those traits, a group of true breeding recessive ones, again in separate corners of his pea plant garden. Then he crossed them, same as he did before, but now he was looking at two traits. When he crossed them, no surprises, right? All the kids, F1 generation, had both dominant traits, round peas and yellow peas, because they got a dominant gene of each type from this parent, right? When he crossed these, this is what told him whether or not these pea color genes had anything to do with the pea shaped genes. And this he got, again, from a ratio in his F2 generation. Just like before, when the 3-to-1 ratio in the F2 generation, when he was looking at just one trait, gave him the idea of pairs and one from each pair, law of segregation. Here it was numbers, different numbers, in the F2 generation that told him what was going on here. Here's a Punnett square to show that. Here we have the basic idea of how all this works, spelled out in all these alphabet letter symbols, right? If I have two parents and they're both heterozygous for two traits, that would be the F1 parents, right? They got a big Y from that parent, they got a little y from that parent. They got a big R from that parent, little r from that parent. If you now cross them, they pass on the genes for these two traits just like we talked about. They're going to give one of the Y's and one of the R's to each of their kids. They're going to give them that by having the formation of gametes, egg cells and sperm cells, with one of each pair in them. So here now, we'll throw this idea in. When you make a Punnett square like is made here, when you put alphabet letters across the top and down one side, this one's kind of tilted horizontally, or whatever you call that. What you are doing is simulating meiosis. You're simulating the actual physical process of cell division we call meiosis, because you are writing down a combination of one gene from each pair, and that is what gametes get. And then when you fill in these boxes in the Punnett square, you are simulating fertilization and the formation of a new individual, and you're simply charting what genes they got. Now, if you take a close look at all those Y's and R's there, you see that if you do this series of crosses, cross the parentals, then cross the F1s to get F2s, in the F2 generation, you'll get 9 offspring that have both dominant traits, in this case yellow and round. You'll get 3 that have one dominant trait, round, and the other recessive, green. You'll get 3 that have the other trait dominant, yellow is dominant, wrinkled recessive. And you'll get 1 that has both recessive traits. This is what you will get, but only if the P-colored genes, the Ys, have nothing to do with the P-shaped genes. Because if they do, you won't get these numbers. Since Mendel did a bunch of these crosses and he always got these numbers, he came up with the idea that when 2 different kinds of genes separate, and here he used the word assortment, when they separate and then go to gametes, assort, they are independent of each other. How a pea plant passes down its pea-colored genes has no effect on how it passes down its pea-shaped genes or its flower-colored genes or other kinds of genes. That's what the idea of independent assortment means. And then we can do this. We can show once again what happens in meiosis to cause this to come out this way. What happens in meiosis, which Mendel would have been totally unaware of, to cause an individual that has a chromosome with a big Y gene and a chromosome with a little y gene and another chromosome with a big P gene, or excuse me, sorry, they use G's, big G, and another chromosome with a little g. These are the F1s, right? I'm now diagramming the chromosomes they have in their cells with those Y and G genes, right? When they make gametes, here's how it works, right? These are going to double. So now we have 2 big Y's, 2 little Y's, 2 big G's, 2 little G's. But when they're finished meiosis and separating, right, these homologous chromosomes are going to separate. And then the chromatids are going to separate. And you're going to get a strand with the big Y in the same cell as a chromosome, a strand with the big G. Why did they end up together? Because they lined up on the same side. And they got pulled apart and put into one cell by meiosis 1. And then you're going to get cells that have a chromosome with a little Y gene on it and a chromosome with a little G. Why? Because they lined up on the same side. So if this was one cell with these genes and chromosomes and they lined up this way, it would make gametes with that, big Y, big G, or gametes with this. However, in the cell next door, when this chromosome with the big Y's lined up like that, and the chromosome with the little Y's like that, if the chromosomes with the G's lined up the other way, the chromosome with the little G's lined up on the left side here, on the same side as the ones with the big Y, and the chromosome with the big G's lined up over here. In other words, they switched places compared to what happened in this cell. In this cell now, they lined up like this. When these get pulled apart, now the chromosome with the big Y is going in the same place with the chromosome with the little g, and you'll end up making a gamete with a big Y and a little g. And another gamete over here with a little y and a big G. And these pea plants make lots of gametes. And in every cell, the lining up of homologous chromosomes, this would be in metaphase 1, right? The lining up of homologous chromosomes is completely random. It's just as likely they line up this way as they line up that way, which means you'll get just as many of each of these 4 kinds of gametes. Not only can each of those 4 types of gametes form, but they'll form in equal numbers when you have a whole bunch of cells doing meiosis. This lining up in metaphase 1 of meiosis, this random lining up of chromosome pairs is the cellular basis of the law of independent assortment. And it's just remarkable that Mendel knew nothing about meiosis, and no one did at the time, and he came up basically with the concepts and the ideas of what happens with chromosomes, because it resulted in those numbers that we showed before, 9 to 3 to 3 to 1. Those would only happen if you did the crosses and things worked this way. The Y genes on that set of chromosomes, on that pair of chromosomes, not set, excuse me, on that pair of chromosomes was independent of the G genes on this pair of chromosomes, independent in what way? In the way they lined up in meiosis 1.